This is a production of Cornell University.
Thank you, everyone. I hope you can hear me all right. So I'm going to jump right into introducing my dissertation topic on copper uptake by grasses at the plant-soil interface. So copper is an essential plant micronutrient, and it plays an important role in photosynthesis, respiration, and reproduction in plants. Now, plants get their copper from soil, and as you can see from this map of the US, copper is naturally present in soils, but this is total concentration. And total concentration is not the same as availability, as we know, because soil conditions change availability of nutrients in soil. So here we can see that copper is absorbed in higher concentrations to these soil constituents as we move up in pH. So specifically, copper absorbs to iron oxides in soil. It also forms complexes with organic matter. This severely limits the availability of copper in soil, and this is particularly problematic in this pH range 6 to about 7.5, which we know is what plants prefer in agricultural systems. And this limitation of copper availability in soils severely decreases agricultural production in these deficient soils, particularly soils with high organic matter content. So as was shown by the Vatamaniuk lab in 2021, wheat is here, and the model species for wheat, Brachypodium distachyon, which I work on, are both very susceptible to copper deficiency. When copper is at low concentrations in their media, they struggle to produce grain. Interestingly, at low concentrations, both wheat and Brachypodium show not a huge difference in the amount of flowers that they produce, but they do produce significantly fewer seeds. What this means for a farmer in a copper deficient soil is you could go out into your field and see tons of wheat flowering and think, I'm going to have a great harvest. You go out there, in reality, if you have copper deficiency, you could have low seed set. This is an issue for food security because wheat requires sufficient copper to produce grain. Wheat is a member of the Poaceae family, or the grass family, which includes the key grain-producing species in agriculture. And the mechanism of copper uptake in these grasses, as indicated by my title, has not been defined. So this is the question that I've dedicated my dissertation to. What is the copper uptake mechanism in grasses? In order to look at this question, I'm starting with the knowledge we already have. So for dicots, or non-grasses, copper is taken up by first being reduced in the soil. So copper in the soil is oxidized, it gets this extra electron, it's reduced, and now it's available for uptake by non-grasses. The same mechanism is required for iron uptake in non-grass plants. So iron has to be reduced before it's available for uptake in non-grasses. The grasses, interestingly, have a different mechanism. They take up oxidized iron. We don't know how they take up copper, but for the iron to be taken up, it must first be chelated before it is available for grasses to access in the soil. In my dissertation, I am spending my first 2 chapters looking at the mechanisms that I just discussed. So considering the reduction mechanism and its efficiency for copper uptake in grasses, same deal for the chelation-based mechanism. And in my 3rd chapter, I'm going to look at these mechanisms in a soil system and consider how soil conditions impact the function of these uptake mechanisms in grasses. So the objective of my first chapter is to establish the copper deficiency response in Brachypodium, the model grass species I'm using, via the reduction-based uptake mechanism. So this is the reduction uptake mechanism in non-grass plants. So in order for copper and iron, here it's showing iron, same for both, to be taken up by the reduction-based mechanism in non-grasses, the plant which is on the left side here, this is the inside of the plant cell, this is the plasma membrane, cell wall, this is the soil. And here we have the oxidized iron in the soil. So the plant first has to solubilize this oxidized iron. It does this by producing protons and potentially phenolic compounds that mobilize oxidized iron in the soil so that it can move into the cell wall and interact with this plasma membrane-bound ferric reductase oxygenase enzyme. This Fro enzyme gives that oxidized metal the electron it needs, so now it can be taken up by high-affinity transporters and brought into the root cell. As I mentioned before, this uptake pathway functions for both iron and copper uptake, but you may note that in this model with Arabidopsis, these Fro genes are somewhat different. So these are different Fro genes for the different metals and different high-affinity transporters for the different metals. They're very specific in each of these uptake mechanisms. Now, looking at Arabidopsis as an example of a non-grass species, Arabidopsis expresses 8 of these Fro genes, whereas Brachypodium expresses only 2. This is one of the reasons that we hypothesize that the reduction-based mechanism is not the primary copper uptake mechanism in grasses and will not respond strongly to copper deficiency. To investigate this hypothesis, I'm cultivating Brachypodium in deep water hydroponics. Doing this because during the last week of cultivation, I can completely deprive these plants of these nutrients and look at their response. So I'm growing them with 3 treatments. I have a control. I have no copper in the system for the last week and no iron in the system for the last week. After these plants are harvested, I have a 2-pronged approach to analyzing the reduction mechanism. So first, I sever the plant roots from the shoot tissues. They're then incubated in solutions that contain chromophores. These solutions develop color over time based on the amount of metals the roots are reducing. I take an aliquot after a specific amount of time of this solution and measure it using UV-Vis, which can allow me to calculate the concentration of metals reduced by the roots using a rearrangement of the Beer-Lambert law. My other method of analysis is to evaluate the transcriptional expression of the ferric reductase genes in Brachypodium. So first I separate the root and shoot tissues, and then I'm analyzing both root and shoot tissues for the 2 ferric reductase enzymes I mentioned previously in Brachypodium using RT-qPCR. These are the results from the reduction mechanism from my preliminary trials. These are reported in a rate. So this is an amount of metal that is reduced per root weight over time. On the left, you have your copper reduction. On the right, there's iron reduction and all 3 treatments. And what we can basically say from this data is that the plants that are experiencing mineral deficiency are not actively reducing more metals in either the copper or the iron assay compared with the control. My transcriptional results show that the Brachypodium Fro1 gene is really not being expressed in the root tissues, but the Brachypodium Fro2 gene is expressed. There isn't a significant difference in the expression of this enzyme between the control and the minus copper conditions, but there is a significant difference between the control and the minus iron conditions. Plants grown in minus iron conditions are expressing significantly less of this enzyme. So taken together, the reduction response data and the transcriptional data support each other to show that in the roots of Brachypodium, this reduction response is not responding strongly to copper or iron deficiency. Interestingly, what we found in the shoot tissue is a more dynamic response to these deficiencies in photosynthetic tissues. So Brachypodium Fro1 is expressed in shoot tissues. It's expressed with increasing, the transcripts have increasing expression under minus iron conditions and possibly minus copper, although my sample set's a bit small. And Brachypodium Fro2 is expressed at lower rates compared to control when plants experience deficiency. We think that this has something to do with the plants trying to relocate the essential nutrients that they have in their photosynthetic tissues when they're experiencing deficiency. They're moving them into photosynthesizing tissues to ensure that they can continue to photosynthesize under deficiency conditions. Returning to the roots, which is what we really care about here right now, my preliminary results indicate that Brachypodium distachyon does not respond strongly to copper deficiency with this reduction-based mechanism, and for that reason, we believe it is not the primary uptake mechanism in grasses. This allows me to move to my second chapter, where I'm exploring the chelation-based mechanism. Less is known about this mechanism, so I have two objectives here. I first want to determine the capacity of associated transporters in this mechanism to move copper across the plasma membrane. Then I want to analyze the response of the chelate-based uptake mechanism to copper deficiency in these grasses. I'm again using Brachypodium. So here's a more detailed explanation of the chelation-based mechanism. On the left this time, you have the soil with this insoluble iron, and on the right, you have the cytosols inside of the plant root cells. In this mechanism, mugenic acids have to first be synthesized within the plant. In order for that to happen, these enzymes need to be present. And these enzymes are only expressed in grasses. This is a very special mechanism that only grasses have. So they synthesize these mugenic acids, which are seen as this squiggly organic molecule with a zetidine on it. Then this mugenic acid molecule is exported to the rhizosphere through this transporter. It interacts with oxidized metal and creates a chelate complex, wraps around the metal, and then the entire chelate complex is brought back into the root cell by these YS transporters. Our hypothesis in this chapter is that the chelation-based uptake mechanism will facilitate copper attainment and will respond to copper deficiency in Brachypodium. I will again be growing these plants in hydroponic solution so I can induce these deficiencies, but this is my proposed uptake pathway for these grasses. So these mugenic acids are specifically identified as Brachypodium products. These are the ones that this plant makes. These are the specific Brachypodium transporters that are orthologs to the system I just showed. And in my hypothesized pathway, these mugenic acids are facilitating copper uptake through these transporters. And this project was funded by the SNP's Small Grants Program in collaboration with Patty Leva. Very grateful for that opportunity. So the methods of analysis after these plants are cultivated in hydroponic solutions, I'll be starting by looking at the mugenic acid quantity for those particular compounds I just showed you that will be collected from roots. They will be quantified using high-performance liquid chromatography mass spec. I will then use the same method I described in the previous chapter, RT-qPCR, but in this case, I'm going to be quantifying the expression of the two transporters that I just showed you that are specific to this system. Sorry about the microphone. Finally, the really cool part about the collaboration, we are going to heterologously express this BDYS1 transporter in an oocyte system and use electrophysiological techniques to establish the capacity of this transporter to move copper through a plasma membrane. I'll be doing this through my collaboration with the Pinedos Lab and Patty Leva. Moving on to chapter 3, where I'm going to be looking at the impact of soil characteristics on copper uptake mechanisms. Specifically, I want to think about how pH, microbial community dynamics, soil organic matter, and mineralogy are impacting the efficiency of these uptake mechanisms in grasses. My hypothesis here is that the grasses are not expressing one or another mechanism. They have all of these genes. I believe they're expressing multiple mechanisms, but that they are able to rely on the mechanism best suited to their surroundings. This improves the efficiency and allows, in this case, Brachypodium distachyon, to utilize the most efficient mechanism for copper uptake based on its surroundings. order to evaluate this hypothesis, I needed to take my hydroponic system to a more realistic level. So I started off with soil. I collected a ton of soil samples that were being cultivated from fields that were cultivated by the Sorrells Wheat Lab, where they are growing a mix of wheat, barley, and rye. Out of the soils that I collected and analyzed, I chose 3. I chose the Caldwell and McGowan soils, which are being cultivated in more conventional agricultural methods, and the Freeville Organic Farm, using more organic methods for cultivation. All of these soils are poorly drained. They all are part of the same taxonomy, but they have different estimated soil series, and importantly, they have a wide range of pH. So the pH ranges from 6.5 to 7.9 in these soils. You can see it on this side too. So this difference is one of the things that I wanted to evaluate in these different soils. I also performed elemental analysis on these soils using ICP-MS. What I'm showing here is total iron and total alumina concentrations in these 3 soils, which are significantly different, indicating they may have differences in mineralogy, but we still need to establish that. I also did loss on ignition to estimate total soil organic matter percent, and I did find some significant differences. So Freeville had higher concentrations of soil organic matter, which could be related to the management techniques. Interestingly, I did not find a lot of difference in the total copper concentration, and this is actually good for me because I want to try to move my system into something more realistic. So I want to add copper to some of these soils and see how the plants respond under these different conditions. In order to do that, I synthesized a mineral copper oxide nanoparticle amendment. So these are just really tiny tenorite minerals. They are an average diameter of 9 to 11 nanometers, which was confirmed via XRD and TEM. And they are a confirmed monoclinic tenorite product. So I want to apply these nanoparticles in the soils, but I needed to know, can the plants actually get to this copper in this mineral? So I started doing some trials with the nanoparticles. I applied them in Cornell mix and grew plants in the Weil Hall growth chamber. I specifically grew a copper-deficient mutant plant that was developed in the Vatamaniuk lab. This is really sensitive to low copper concentrations. And this plant in the typical lab practice is fertilized with copper sulfate, so just a little solution as opposed to a mineral. I use this copper sulfate as a control treatment. And then I added different concentrations of copper oxide nanoparticles. And I found that the flowering response was rescued in this mutant above about 2 parts per million copper oxide nanoparticle amendment. But that didn't really tell me where the copper was going, either in the soil or in the plants. So I wanted to understand distribution. In order to do that, I went over to the Cornell High Energy Synchrotron facility, where we carried out a collaboration using these rhizoboxes to live image plants interacting with the nanoparticle treated soil. And these are the results from that experiment. So down here on the bottom, we'll look at this one. Down here on the bottom, we have a 0 treatment here, so no addition of copper. This is our control. And then the 3 in the middle are copper oxide nanoparticle treated soils. This is the same order that you see here. And you can see that as you increase the treatment of nanoparticles, there's more accumulation of copper, not just in the soil, but in the actual root tissue. What I found was that it wasn't just the root tissue, it was also the shoot tissue. Moving up in concentration of treatment here, these are leaves. These stripes are the actual vasculature within those leaves. So we're seeing higher concentrations of copper in the leaves of plants that were treated with these copper oxide nanoparticles. So this confirms the idea that the nanoparticles were not only available to the plants, but that they were getting the copper out and moving it through the plant tissues. I will also be using in this chapter of my dissertation, a really important technique of multi-collector ICP-MS. I won't bore you with all the details, but essentially this allows us to distinguish the primary copper uptake mechanism by analyzing plant tissues. We can analyze these plant tissues for the specific stable isotope ratio for copper, which doesn't change in a chelation-based system, but if the plant is primarily using a reduction-based mechanism for copper uptake, the reduction prefers the lighter isotope. So that changes the isotope ratio in the plant tissue and gives us a metric where we can look at which mechanism is actually being favored based just off those copper isotopes. This will be very important for this chapter. Other methods of analysis I plan to use, I want to look at the microbial community dynamics in these soils using 16S amplicon sequencing. I'll use RT-qPCR, metabolite analysis, and the reductase assays mentioned previously to analyze the response of those particular mechanisms. So in summary, if you take nothing else away from this talk, copper is essential and we don't know how it gets into grasses. In my dissertation, I'm going to be looking at 2 established fundamental uptake mechanisms and determining if they are functioning in these grasses, if they are what's bringing the copper in. And the response of copper uptake mechanisms to different soil conditions is going to be evaluated in pot studies using field soil as an attempt to apply these fundamental concepts in a more realistic way. Finally, I want to thank my committee. It has been so incredible to be co-advised and get to be part of 2 amazing labs. It's been great for my interdisciplinary experience. And of course, thanks to all the people on this slide and my critters and my family. And finally, please check this out. This is the new XLEAP project at the Cornell High Energy Synchrotron. There are amazing opportunities for biologists, for soil scientists. This is going to be great for us. So take a look at that. Thank you.
This has been a production of Cornell University, on the web at cornell.edu.